Okay, so thank you everybody for joining us today. So we have a really different type of discussion that we wanted to have today versus our usual type of presentation. So in this issue of our webinar series, we're going to have a fireside chat with Marco Vaccaro and Patrick Bajoro, first authors of the recently published manuscript Spatial Multimodal Analysis of Transcriptomes and Metabolomes of Tissue in Nature Biotechnology. Marco and Patrick work in the laboratories of Per Andren and Joachim Lundenberg. So, what I really like about this work is it represents a multidisciplinary team investigating different molecular levels as well as combining different approaches. So, what we're going to do first is we're going to have around a 35-minute team presentation of data related to the study. We're going to do an interview on the study and then follow this with a 15-minute Q&A session from the audience. So as a brief introduction, I am Peggy Angel, who's hosting this meeting, and Kristen Burnham Johnson will be presenting the two authors of the paper. Kristen?
Hi, everyone. So as Peggy stated, I'm Kristen Burnham Johnson. Thank you so much, everyone, for attending today's talk. It will be great. So we have Patrick, but to kick it off, we're going to have Marco go ahead and present his portion first.
I was muted, sorry. Hi, everyone. Thank you, Peggy and Christine, for inviting us. And let me share my screen. Okay, so as Peggy said, we will present our work that is named Spatial Multimodal Analysis of Transcriptomes and Metabolomes in Tissues. And I will focus on the experimental part and especially on the spatial transcriptomics part, whereas Patrick will focus on the multi-MSI part. So just a little note before I move on to the presentation. I might use the multimodal and multiome terms interchangeably, and also spatially resolved transcriptomics and spatial transcriptomics. So whenever I use these terms, please assume that I, like for the sake of this presentation, I mean the very same. So first of all, why multimodality? So we have very good technologies in our labs that are able to profile either single molecules or also many molecules together up to an entire class of molecules. But biological processes happen at many biological levels. So if we really want a deep understanding into biological processes, it's necessary to be able to profile several classes of molecules together. But then maybe we can ask, why spatial multimodality? Well, an easy answer is that not all the biological processes happen inside the single cells, but they happen also between the cells and their environment or between cells. So like for the first aim, like profiling biological processes into single cells, there are already a plethora of methods that have been developed. Whereas like for the second aim, you could use spatial multimodal or multiome methods. But there is also a second reason, and the reason is that with spatial multimodal methods, many times it's much easier to combine different technologies that they have different readouts. So for example, here I'm highlighting the possibility of profiling also the proteome, not only using like a common readout between antibodies and proteins like with DNA, but also with different readouts. And here is an example with fluorescent antibodies. Now I will focus a little bit more on what is spatially resolved transcriptomics. This is an umbrella term that it's about all the technologies that have been developed to enable us to study the RNA in its native localization. And as you can see from this timeline, many technologies are quite quite old, like from the 80s. But it's only like in 2016 that the first technology that enables the profiling of the RNA using next generation sequencing was developed, that the field got a lot of attention. And this technology received the Nature Method of the Year 2020 award. And after that, this was acquired by 10x Genomics, and this ST technology was turned into an enhanced and much more powerful technique, which is called 10x Visium now. In blue, you can see that I'm highlighting also ISS, because in this work, we have actually used both techniques together. So what is Visium, and like how does it work? So Visium is a technology that enables the profiling of virtually the whole transcriptome, but it has a resolution that is not single cell resolution. So it's about it's 100 micron resolution. And it works by placing a thin tissue section on these little arrays, which are covered with millions of probes, which have a unique spatial barcode, and they can capture the polyadenylated RNA. So once the mRNA is captured, it can be reverse transcribed, and then the cDNA, which will contain also the spatial barcodes, it can be amplified and then processed into an library that can be sequenced with Illumina sequencers. Whereas ISS, it's a different technology that enables the profiling only of hundreds of genes, but it has a much higher resolution. So it can go into subcellular resolution. And the reason why it can do that is that it uses padlock probes, which then after ligation, they can get Rollin circle amplified. And this enhances a lot the signal, which can be read using sequencing by ligation. So now we can get into our work. And as I said, I will focus on the part about spatial transcriptomics. So we aimed to apply multi-MSI and spatial transcriptomics to the very same tissue section. So as you can imagine, there were some obstacles that we had to like overcome. There are some obstacles that are specific to multi-MSI, because Visium can be performed, or at least at the time could be performed only on Visium arrays, which are non-conductive. And Patrick and Reza will focus on this part. Whereas the other obstacle can be that, of course, if you do multi-MSI before Visium, there is a high chance that maybe we didn't know how could it be the quality of the RNA afterwards. So I will focus more on this second question. And here I'm showing you the first pilot experiment that we did. So we cut 8 mouse brain tissue section on a Visium array that contains 8 arrays and 8 capture areas and not 4. And this is an array that can be used just to check the quality of the RNA, because this kind of experiment doesn't end up in a real Visium sequencing experiment, whereas you just perform a reverse transcription using fluorescent nucleotides, and then you just get a fluorescent signal. So we sprayed on this array 4 different MALDI matrices, DHB, Norarmain, 9AA, and FMP10. And then, surprisingly, we found that not only we could get a good MSI signal that you can see in B, but also that, as you can see in C, the RNA was still present after MALDI-MSI. And then we performed also a confirmatory experiment using ISS that you can see in D, where a single tissue section was sprayed with FMP10, and then we imaged only one side of the brain, and we used the other part as a control. And you can see that like the rolling circle products signals, they are very, very similar in both sides of the brain. So this first pilot experiment really motivated us into going into real sequencing experiments that I will show you in a minute. But before doing that, I just want to mention, like, how do we usually analyze our data? So a Visium sequencing experiments ends up in a matrix, a count matrix, where you have all the genes that have been detected in each and every spot of the Visium array. So here you can see some methods that have been developed for single-cell RNA sequencing. And the reason is that the spatial field, it's kind of like it has borrowed many methods that have been developed already for single-cell RNA-seq. And one pipeline that we commonly use to analyze our data is Seurat, where after normalization, we select the highly variable genes, and then we also select a number of principal components that we are interested in. And we can use this to infer a shared nearest neighbors graph that can be used to cluster our data. And we can reduce the dimensionality of our data using t-SNEs or UMAPs. And this kind of pipeline that initially was developed for single-cell RNA-seq data, now it has been commonly applied also to spatial transcriptomics data, as you can see from these screenshots that are taken from the Seurat website. And this is exactly the kind of analysis that we use with our data. And as you can see in the in the first 2 UMAPs that I'm presenting, the colors indicate the technical conditions, and the shapes of the spots, they indicate the replicates. And I know that it's very difficult to distinguish the colors and the shapes, but this is exactly the purpose of the image, because we want to show that regardless of the technical condition or the biological replicates, the spots are not the clouds of spots do not they perfectly overlap. Whereas if you cluster your data, then the results of your clustering, they are actually clusters that represent meaningful regions in the mouse brain, as you can see in the spatial plots below. And they are very, very similar regardless of the matrix that had been sprayed on the sections before the analysis, or if it is like a section that has been analyzed with standard Visium. At this point, we wanted to apply this technology to a real biological question. And for this, we chose 6-OHDA mouse models that are mice that have been injected with this toxin, 6-OHDA, which kills the dopaminergic neurons. But since they have been injected unilaterally, this will kill the dopaminergic neurons only on one side of the brain. So using the multi-MSI component of our multimodal technology, we were able to reliably detect the dopamine only on the left side of the brain. And then using the spatial transcriptomics component, we were able to detect the genes that are most highly correlated to dopamine. But also another thing that you can do with spatial transcriptomics data is inferring the cell type proportions that are inside each spot. And doing this kind of inference, we could interestingly detect exactly the dopaminergic neurons that are mostly affected by the lesion. Finally, we wanted to apply the very same strategy to a human postmortem sample. So this sample had been in our -80 freezers for more than 6 years. So we were expecting that the RNA was very highly degraded. And for this reason, we decided to apply a protocol that has been recently developed by our lab just to analyze samples with highly degraded RNA, which is cited below. And it gave, like this kind of analysis, it gave very similar results. And we could once again detect the genes that are highly correlated to dopamine, and then infer the cell types. And once again, we found very enriched in the region of the striatum where dopamine is still present, this population of medium spiny neurons, which are contacted by the dopaminergic neurons in this area of the brain. And with this, I thank you very much for the attention, and I give the word to Patrick.
Thank you, Marco. Okay. Let's see here. Okay, let's talk a bit more about the mass spectrometry imaging part of the study. Okay, so there are different ways of doing mass spectrometry imaging. The one we use in our lab is called MALDI, which is matrix-assisted laser desorption ionization mass spectrometry imaging. So the clues there is in the name. So we use a matrix to get the ions out of the tissue, which then separates in the mass spectrometer, and we detect with the mass spectrometer. So one thing about MALDI is that you require chemical matrices to make the ions. So I've listed 3 here. There are definitely more, but these are 3 of the main ones that we did for this this paper. And we don't require too much equipment for MSI. We have a sprayer, which we use to get the matrix on the tissue. We have a mass spectrometer. In this case, it was a 7 Tesla FTICR from Bruker, and the software that we use to look at the data is called Skills, which is also from Bruker. So a typical mass spectrometry imaging workflow usually starts with some kind of tissue. In this case, it was a brain, mouse brain to be precise. So you start by cutting it using cryomicrotome, which means that you have we have fresh frozen samples. We then do the MALDI. We apply the matrix using the sprayer that we can see here in the image on top, and we analyze it using the mass spectrometer. The difference here is that when we also want to do spatial transcriptomics afterwards, we remove the matrix before that. So there is no interaction going on there. So as I mentioned before, we have 3 different matrices that we chose for this paper. We have FMP10, which is a derivatizing matrix that was developed in our lab a few years ago. We also have 9A, which makes negative ions, and DHB, which makes positive ions. So one reason of using 3 different matrices is to cover more things that we are able to see. So one thing about FMP10 is that it derivatizes, so it actually binds to the molecule that we want to look at, but it's specific to phenolic hydroxyls and primary amines. So mostly we see is neurotransmitters and amino acids. And so we use FMP10 to find metabolites that are difficult to ionize and also low abundance metabolites. For 9A, what we use it to see is mostly amino acids. We see the energy metabolites and also small molecules. And for DHB, we see lipids, small molecules, and peptides. So this is one of the images from a paper we did, or our lab did in 2019. So that was the paper that described FMP10. So one reason why I wanted to show this is that it's going to be relevant also because we did a similar thing now for this paper. So like Marco was saying before, we have this 6-OHDA neurotoxin that is injected unilaterally, meaning that we have half of the brain with Parkinsonian symptoms and half of the brain is intact. So we have case and control in the same tissue. And as we can see here, we have a few metabolites and neurotransmitters with unilateral distribution, and then some that are not affected by the neurotoxins, which seem to have a bilateral distribution. So one thing to keep in mind then for the data analysis is you have different masses that you have to look at. So with 9A and DHB, it's more people are used to the math there that goes into identifying a peak. So dopamine, for example, with 9A, you would see at 152 because it loses a hydrogen. DHB, you might see it at 154 because it gains a hydrogen. But with FMP10, we actually have FMP10 binding to the molecules, like I said before, at either the phenolic hydroxyls or the primary amine. And yet another thing you have to keep in mind is that this can also happen doubly. So you can have 2 FMP10 molecules attaching to dopamine. So instead of finding it at 152 or 154, you would find dopamine at 421 when it's single derivatized by FMP10. So it's instead a delta mass over charge of 268. But one thing that we keep in mind when it's moldy is that you only have single charged ions, so we don't see any double or triple charged ions. Okay, so moving on now more to the paper that we are here to discuss, we have spatial multimodal analysis. And of course, I'm going to talk more about the moldy MSI parts. So here, one of the biggest things that we wanted to look at is because a moldy MSI is done before spatial transcriptomics, the big difference here compared to regular MSI is that we were doing the experiments on Visium glass slides, which are non-conductive. And in a regular MSI experiment, we would use conductive glass slides. So here I would like my colleague Reza to explain a bit more.
Thank you, Patrick. There are 2 challenges for doing a spatial multimodal analysis like spatial transcriptomics after moldy MSI. First, moldy MSI is a destructive technique to some extent, meaning that laser beam will ablate the tissue to generate ions. So the first question is if the integrity of the tissue and quality of RNA is preserved for following transcriptomics analysis. Here you see the brain tissue section coated by moldy matrix, which is Norharman here in this case, and is running inside the Solarix FTICR instrument. And as you see, there are obvious signs of major ablation of the tissue. As you have seen already in our result, it's proved that the sample after MSI analysis and washing of the matrix has good enough quality for following transcriptomics analysis. And the second challenge is that traditionally conductive glass slides like indium tin oxide coated glass are used for mold imaging. And that's to avoid the charging effect and fading the signal over the time. While the following mode analysis, for example, transcriptomics, uses Visium slide, which is not conductive. So using our new generation mass spectrometers, like timsTOF and Solaris FTICR instrument, we compared non-conductive superfrost slide with conductive ITO glass slide and coated both with FMP10 for imaging of neurotransmitters. And as you can see here, the signal for dopamine, which is about the same on both on superfrost and on ITO glass slide. Thank you, Patrick. So please go on.
Yes. So like I said before, we're going to come back to this image of the 6-OHDA lesioned mouse. So this is a similar image that we did now for this project, but the difference being that this is on Visium glass slides, so non-conductive glass slides. And we still can see the unilateral pattern here of having dopamine, norepinephrine, DOPAC, 3-MT, unilaterally distributed, with one side of the brain being control and then one side being showing Parkinsonian symptoms, which is very promising. So as Marco mentioned, also we did some human data as well, just to see that this also was working okay for other modalities. So we have about a 9.5 hour post-mortem time, but we can still see quite a few of the Parkinsonian metabolites. So on the right here, we have, for example, dopamine, we have 3-MT, serotonin, and norepinephrine. Norepinephrine being double derivatized here. So this was also done in FMP10, and in this, I should mention also that we collected the samples based on the Visium squares that we can see here on the left. So that's those green squares are where Visium collects the RNA data, and so that's where we collected the MSI data as well. So we wanted to put some numbers also to how well SMA MSI, so non-conductive glass slide MSI, compares to regular MSI, meaning ITO or conductive glass slides. And as we expected, similar glass slides, so SMA here will, we mean non-conductive glass slides, and MSI that we have down here is conductive glass slides. And so as we expected, we saw an almost perfect correlation between samples on the same type of glass. For FMP10, we saw about 0.85 Pearson correlation between conductive and non-conductive glasses. Moving over instead to DHB, we saw still here an almost perfect correlation between the same glass slides and 0.74 to 0.77 correlation for different glasses. And a similar thing then also for 9AA, where we had almost perfect correlation for the same glass and slightly lower for different glasses. So looking at this a different way, we also wanted to plot the intensity of a given peak list for the same three matrices. So on the top half here, we can see intensities plotted for non-conductive and conductive glass slides. And for FMP10, we have almost 96% of all the peaks that fall within half a log 10 fold change between them. And for DHB, that's almost 92%. And for 9EA, we had slightly lower of 61.6% of them falling within half a log 10 fold change. And we also wanted to look at some data analysis. Marco showed some clustering. Here we have a 10K nearest neighbors plot where there was a joint dimensionality reduction, and then the clustering is based on cosine similarity. One thing we can see here in the C panel is everything that was, or genes and peaks that were associated with dopamine, so double derivatized dopamine. And one thing that was very promising is also that we can see the single derivatized dopamine mass that we remember from the beginning of the talk. So to conclude this, we show spatial multimodal analysis of metabolites and mRNAs in the very same tissue. We also show that MALDI-MSI can be performed on non-conductive Visium glass slides, and that there is no significant loss of mRNA after MALDI-MSI on the same tissue, which enables spatial transcriptomics. So with that, I would like to thank you on behalf of all of us, and thank you for listening, and thank you for inviting us.
Thank you so much, Patrick and Marco. Those were beautiful talks, beautifully explaining the entire study. So what I want to move on to now is just a little bit of a chat with you, discussing certain aspects of the study. And Kristen and I are going to ask different questions, and then the audience, please put your questions in the Q&A chat, and we will take questions after we get finished with the interview. So one of the things I really like about this study, and one of the things I like about imaging mass spectrometry in general, is that it is very multidisciplinary. So you have people from different teams coming together with their expertise. So in this case, what I wonder is, how did the two groups get acquainted? And then how did each of you step forward and take the lead on certain parts of these studies? Maybe a little bit about your background and how you came to your positions, and maybe a little description of how the two groups started collaborating together.
Okay, so I'm a medical doctor, and I'm a specialist in clinical pathology and clinical biochemistry, it's called. And I really wanted to do a PhD in biotechnology, working with developing new technologies. And this is the reason why I applied for a position at the lab where I'm at now. And the reason why the two of us got acquainted is that we work at this biotechnology infrastructure that is called SciLab Lab, where within many other things there is also a spatial platform where all the all the groups that work with spatial technologies are part of. And spatial transcriptomics, it's one unit of the platform. MALDI-MSI another one. ISS, it's also another one. So basically that helped a lot, like to get in touch and start a project.
Yeah,
really...
so my background is not a medical doctor, but my background is in biomedicine. And over the course of me studying, I became more and more interested in bioinformatics. And so when I did my master's project with Lucas Schell, who's also on the paper, he mentioned Per Andrén's lab, and set me up with that contact. And I've been there since 2020 now.
Great. I'll go ahead and take the second question for you guys, and both of you can address it. So you guys had a lot of firsts in this paper. And in your opinion, what ideas and concepts or past literature really led to this elevated collaborative study?
So I think one of the biggest things was maybe more a gap in literature, that we have the infrastructures to do both techniques. We have the infrastructure SciLife lab that Marco was talking about. So it was everyone was very fond of us collaborating. So that's ended very well, or it started very well. So should say.
Great.
Yeah, like from a literature point of view, I think that one work that was very important for us is the one that Patrick cited, and it's about using this derivatization matrix FMP10 to be able to detect neurotransmitters. So that was very important, like also to target some real biological questions. Whereas like from our spatial transcriptomics point of view, it was more about the fact that now it's a really hot topic, like developing technologies that can profile the epigenome and the molecular environment together. And I was briefly mentioning this during the talk that it was possible to integrate proteomics inside single-cell RNA sequencing techniques after the invention of these DNA-tagged antibodies. But the metabolome, somehow it's still kind of difficult, like to integrate, especially with droplet-based assays. And this is why I mentioned that maybe we figured out that probably using the spatial coordinates, it could be somehow easier to integrate the two layers of information, given that the RNA could still be detectable or at least analyzable. So I think this was the background that led to the work.
Great. Thank you. Do you want to take the next one, Peggy?
Yeah, thanks. So one of the things I wondered is, as you were talking about all these technical details that I really love discussing, like the slide conductivity and comparison between the slides, what was the biggest hurdle in combining the metabolomics and transcriptomics? And how did you overcome this hurdle?
So like from strangely enough, from the point of view of like producing the data, it was actually kind of straightforward. Like we just like did like some hypothesis of like how we could combine the two techniques, and then we just tried. But fun enough, like something that we struggled and we are still somehow struggling, it's really integrating the two data sets. Yeah, it's something that like maybe Patrick can say something more about that, but it really refers to like the kind of tools that we can use to do this alignment and then integration of the data.
Yeah, exactly. So, I mean, at the moment, spatial transcriptomics and MSI have all been developed to fit into their own areas. But I think we need to think a lot about how to integrate these, sorry, both for aligning images, for example, and aligning data and being sure that we can compare them in a fair way. So I think that's definitely one of the hurdles that we're working on and will be working on.
Great, thank you so much. One of the things I think I was surprised about is that there seemed, and hopefully I'm not misinterpreting something, but it seemed that there was a really good correlation between some of the transcriptomics and metabolomics. We don't always see that at the protein level. Is this something that was expected? Or maybe I'm misinterpreting the data.
Do you want to take that one?
Yeah, can you repeat the question? Sorry, Peggy.
So what I wanted to know is, so usually when we look at transcriptomics, just from a comprehensive viewpoint, for instance, not even spatial, and compare it to proteomics, there can be timing differences, right?
Yeah.
So one of the things that I thought was interesting was that it seemed that the metabolomics, and again, I could be misinterpreting the paper, the metabolomics and the transcriptomics seemed like they were closer in timing compared. It really kind of surprised me in that.
Mm-hmm. Yeah, that's a very interesting question. And actually, I think that this
Mhm.
is the aspect that it would be the most interesting, like to actually explore. I must say that like in our paper, we actually we didn't really explore very deeply
Mhm.
the part of like the when I say like metabolome, I mean like glucose and other
Mhm.
like energy metabolites, but we really focused on neurotransmitters.
Mhm.
And we focus on neurotransmitters in like this mouse model where like dopaminergic neurons are already dead, and on a post-mortem sample where like the disease was quite advanced. So this kind of timing thing, like maybe it could be just like a feeling that you get by like taking a picture when like the process is already kind of complete. But
Mhm.
I think it would be super interesting like to dive deeply into like these correlations of like RNA and many other molecules together.
Thank you. Kristen, do you want to take the next question?
I will. And yeah, I think that's a great question too that Peggy brought up, because we do see so much post-transcriptional and post-translational control for proteomics. But yeah, in your system, looking at it for those specific targets, I think it gave you a lot of advantages. But my next question is big and wide open, and really to give your guys' input. So lots of innovation in this. What is the biggest innovation in your opinion for both of you guys?
Doing doing both techniques in the very same tissue, I would say.
Yeah.
Um, you can always do the two different techniques on consecutive tissues, which is good, but then it's, in our opinion, it's better to do it in the same one, and being able to show that we can do this is the biggest thing, I think.
Do you agree, Marco?
Yeah, I totally agree. I
Yeah.
think there is a number of papers like that have been published before us that show that this is perfectly possible, like in consecutive sections. But then at that point, like which I think it's super good, but because like you get like the data quality of the two modalities, it's perfect because you use both technologies in their native kind of environment. But then you're left with the problem that you need to align the data, and so it kind of becomes a computational task. In our case, the advantage is that in order to combine the data, you only would need to put them in the same coordinate framework. But then this can really be used like to uh, be sure that the measurement it's that one, and maybe like even to bridge other data sets that have been taken on single sections. But yeah, I totally agree with Patrick.
Do you want
Okay.
to take the next one, Peggy?
Yeah, thank you.
Mm-hmm.
So, one of the things I wonder is, what do you see as the biggest issue if you took this to a much larger preclinical or clinical study that was combining both the transcriptomic and metabolomics?
Yeah, funding maybe.
This, anything else?
Yeah. So no, like I said it as a joke, but
Yeah.
for real, like I really see that like as a major bottleneck, both from the Visium side, where like we have a spatial transcriptomics side, where there are very good commercially available kits or methods, or on the other side, also on the multi-MSI side or MSI side in general, where like maybe performing like the machines themselves are very, very expensive, but also like the expertise of people that can do this kind of assays, it's kind of rare. So these, I think, they can be limitations for like big big studies.
Is it okay to ask an estimation of the cost for the Visium? That's something that I'm concerned about as well.
Oh, so like now I don't have like the precise numbers at hand, but it's thousands of dollars.
Mhm.
So like the cost of Visium, they are split between the cost of a slide and the
Mm.
costs of sequencing. So even sequencing, like it's getting cheaper and cheaper every time, but it's still kind of expensive. So I think it's in the order of like, how much was that? I think $1,500 per or $1,000 per capture area.
Mm.
So it's like 4 to 6. I can be absolutely wrong, so please don't take my word for it. And then maybe other $2,000 for the sequencing. So probably less than $10,000 for 4 samples.
Mm. So it's maybe not ready, quite ready for moving into preclinical and clinical studies.
Yeah, given the funding, everything is possible. But I don't know, maybe also the computational side can be a huge, like when you produce like such a big data set,
Mm-hmm.
then I don't know, I'm not very expert in computational methods. I don't know what you think about this, Patrick.
Yeah, I mean, doing it manually definitely has a limit. So that's something for the future as well, to work at automating a lot of stuff that can be automated. So yeah, I think we're thinking about cost in different ways. So not just meaning money, but also hours worked and instrument time and so on.
And as you mentioned before, all the pathway integrations. So that was your manual time of putting all the details together. Gotcha.
Kristen, maybe we each ask one more question.
Yeah. We've already touched on a lot of these, but let's go with another broad question. So if you could do the study again, is there anything that you would change?
I don't know, like so even here, I think that the costs of the technologies, it had an impact. So like in an ideal scenario, I would definitely love to do like much more replicates than the ones that we did. Or I don't know, maybe we can still do something more in the future. But yeah, like I would definitely like try to analyze more samples and design in a more balanced way, like the part that we did about the technical benchmarking of the technology. Because I think that you notice that early on we already had in mind that we wanted to try to address some biological questions. So even for the technical analysis, we ended up having like much more much more samples with FMP10 rather than the other matrices that maybe we have not tested like with so many replicates. So yeah, maybe I would redo and possibly expand that part.
Yeah, and before Patrick answers, just one thing that you said that struck me is for the human tissue. You said it was 6 years old at the -80 and the RNA looked great. So that means that there could be a lot of biobanks out there that are available for analyzing older tissue. Because I think a lot of us think as RNA of not holding up that long and that being that high quality. But in your experience, sounds like the older tissue is definitely suitable.
Well, I must say that the RNA quality was not that great.
Okay.
And I mean, if you go to the extended data of the paper, you can find like we published the QCs for this sample. What we found out is that even using like thanks to the use of a technology that in our lab it was developed like to analyze these highly degraded tissues. I think that we managed to put this sample like in kind of like right on the other side of a threshold where it would be completely not possible to analyze. And we managed to extract, I think, like the minimum amount of information that we were looking for. Yes, definitely, like biobanks are our resource. I don't know honestly, like how good the data that we can get from them. Like I expect like to find even like highly like samples with highly degraded RNA, which will not be possible like with the techniques of today to analyze them good enough. What I must say is that we analyzed the same sample like with our multimodal technology. So we had the multi-MSI like modality and then the RNA modality. The very same sample, just like another section, it was already analyzed, I think, 3 years ago in the first paper where we published about FMP10. And then we compared the data, like the multi-data with the previous data. And I believe, I don't know, like Reza or Anna could say otherwise, but I believe that that part of the data that indeed looked very similar. So from a point of neurotransmitters, maybe, or other metabolites, maybe biobanks can really be like a much better resource. But I think that for RNA, it can still be a limitation.
Gotcha. I guess, Patrick, if there's anything that you could change.
Well, I would be on the same path as Marco is. I mean, you always want to do more than you're able to because of different reasons. So, I mean, from the MSI part, it would definitely be interesting to try even more matrices to see what we can find. So yeah, and also, I mean, obviously continuing on the data analysis, but that, I mean, we can still do that.
Right? Find that magic matrix that gives you everything all at once, right?
Exactly.
Yes.
Okay, we're running a little short on time with this wonderful discussion. So I'm going to move to asking some of the audience questions. So Kristen, maybe we can go back and forth on these.
Mm-hmm.
So the first question is, how did the unilateral injection happen?
It's a stereotoxic injection with 3 micrograms of 6-HDA in the median forebrain bundle, only on one side of the brain.
So is it a direct syringe? Is direct needle to the brain?
Yeah, I believe so.
Yeah, very precise, and so it's not us that do it ourselves, but our collaborator, and they have years and years of experience in using these stereotactic devices to find the exact right position in the brain to inject the toxin.
So is it a situation where you have to have some type of opening to access the brain area?
No,
Just
so
curious.
you sedate, I mean, the animal, but then I think it's a very fine needle you inject into the brain.
Okay, thank you. Kristen?
Yeah, for the next ones, why does regular MSI need conductive glass slides?
So, I mean, need is maybe not the best word here, but we usually use conductive glass slides because it yields more ions. We can, as we showed here, use non-conductive glass slides. Maybe Anna Reza wants to add something there.
I can add, Patrick. I think it's because the tissue usually play the role of an insulator, and then during the moldy process, the charge going to be accumulated on the tissue, which affect the ion beam. So what you see as a result is that the beam going to be kind of not really centered, so the result going to be fading the signal. So that's why you need the conductivity to get rid of the formed charge. And why it doesn't happen for us is it's because of the electrical engineering or the geometry of the source of the instrument that we have. Because for the older instrument, like the Flex Extreme that we had a lot of charging effect problems, so it wasn't possible to do imaging on non-conductive slide. But that's the charging effect. Short answer.
Great.
So that leads into the next question really is it possible to use non-conductive slides for multi-Rapiflex imaging and then followed by Visium transcriptomics? Or can we use a Visium slide for Rapiflex imaging? And I think this gets again to the source configuration, Reza.
That's true. Yeah, we really haven't tried Visium on this kind of instrument. We never tried on that. But what we saw on this type of instrument was the fading signal after a while, actually, on non-conductive slides. So yeah, I don't know really. We haven't tried on Visium. Yeah.
When you were comparing the different matrices to each other on the different conductive or the general path slides, did you see a difference in peak intensity versus I know you showed that. Did you see any mass shifts is what I wondered as well with the non-conductive slides?
No, we actually didn't see any mass shift. We have overlapping when we compare the data in skills, both runs, we have the peaks completely overlapping, but they are different in intensity.
Interesting.
Specific, yeah.
Thank you. Kristen?
Okay, I'll take the next one. So this one's more sample prep. So matrix wash with the 70% ethanol followed by the 100% for the slide. Is it okay to do spatial transcriptomics? Will it affect the data?
So like it very much depends on which kind of section or which kind of section are you talking about. So if it is an FFP sample, I think it might work because it's already fixed, and we actually deparaffinize the sample using these solvents. We just do it like the other way around. So we go from ethanol 100% to 70%. But in our case, we used fresh frozen tissue sections, and we wanted to clean the matrix with methanol, because we knew that this was it was like a reagent that is already compatible with the Visium protocol, because in Visium the sample is fixed with methanol fixation using cold methanol. So this was the reason.
Thank you, Marco.
Thank you. So one last question from the audience. Did you mix all the matrices and spray them together, or did you spray them separately one by one? Maybe you can describe why you sprayed each matrix.
So we spray them one by one. So we have one matrix per tissue section. Yeah, the second part of the question, maybe Anna and Reza will be.
No, so yeah, so I mean, we did have one of each matrix on the very same Visium glass slide, but in the different capture areas, because we were aiming to obtain both negative small metabolites, the neurotransmitters, and I mean lipids and other positive metabolites from these three sections at the same time.
Okay, great. Any other final questions or discussion points? No? Okay, I really think this has been a wonderful discussion. I love the format, and I really appreciate everybody from Europe staying up so late to present this data. I think it's a really important paper and a great demonstration of teamwork and combining different modalities. So thank you all so much.
Yeah, thank you, everyone.
Yeah,
Yeah, thank you for inviting us.
Yeah.
Thank you.
thank you. Bye-bye.
Bye.